Hello guys, so let's have part 2 of this plant systematic series. And let's have a closer look at plant identification first, then maybe some data information systems from the internet. Now, when we talk about identification, the first thing that we should do, of course, is to compare an unknown with a known. For example, for any given object, you can say that you have identified it because there's already an existing object that is the same or similar to it that you already know. Now, if there is none to compare, it remains unknown. Okay, not until you tell other people, for example, that you first discovered it and you assign a name to it. Of course, when it becomes known, it will also become the basis of identifying other objects that look the same or similar. Or maybe you can also equate identification with grouping things. Okay, you identify what belongs to a group or what is associated with a group of things that maybe possess unique diagnostic features. Now in plants, we need to set what is known about the taxa being considered. So we basically rely on features called diagnostic characters. If our unknown plant has the same features as our known, then it automatically becomes part of the known taxa. Now, there are many ways or methods to identify plants. Now that you know the basic nature of identifying things, let's look at taxonomic keys, written descriptions, okay, your specimen and image comparisons, and of course your expert determination. All of this utilize known data to be able to identify the unknown at hand. So let's start here with taxonomic keys. Now I have mentioned this already in the first video of the series. And the most common is your dichotomous key. Okay, you can check some textbooks. But here, I'll show you an online version. For example, in Araneta, we have here two leads, okay, known as the couplet. Two contrasting statements. Okay, plants thalloid versus non-thalloid. Okay, and also listed here are the textbook references. Let's show the couplets here. As you can see, guys, this is the key to the family Araneta. Okay, most dichotomous keys are constructed like this because obviously you need a large paper maybe to include all the plant taxa. And not all taxa are well studied or defined like this. So usually dichotomous keys are available only for very well studied taxa with a complete description of all the diagnostic features or characters from maybe roots to flowers to internal sections and the likes. Okay, so let's choose this lead. Then we have an additional two leads here. Let's choose this. Thalli without roots, lacking veins, flowers without a spot. You can check, we have two genus here. You can check if your unknown specimen is the same as this. Okay, so let's have Wolffia. We have here specific examples. For example, Wolffia borealis. And there are several information about the species that you can use to verify your specimen. But just a word of advice, guys, most dichotomous keys like this are made for practical uses. So they are only used to identify. Most do not really reflect phylogeny. So you should be cautious, for example, to say that Wolffia here is closely related to Wolffiella, for example. They are written side by side in a dichotomous key. It doesn't mean that they are related. But of course, for this two genus here, they belong to the duckweed family. But again, it's not always the same to all entries in all dichotomous keys available. Now, according to Simpson, there is what we call as artificial and natural keys. Okay, as the name implies, artificial keys are for practical purposes of identification. Again, most of the useful keys that we know today belong to this category. Then the natural keys more or less tell something about phylogeny. And you rarely find this due to let's say, difficulties at the moment, as there are still perhaps plenty of problematic themes when it comes to phylogenetic classification. Now, when using a dichotomous key, okay, you have to read it and examine it carefully, especially the leads, because most of us jump directly to the next couplet after the first lead seemed to fit. Again, think twice, because the second lead may fit better. Okay, let us now look at another type of key called a polyclave key. As the term implies, this uses a variety of character states. What are these again? Your character states, things like, for example, your flower color, seed shape, fleshy or woody stems, root structure, and many more. Now, what you do is to simply encode all the character states of your specimen and check what matches the most with the known taxa in the database. And I forgot to mention again that this needs some computing power to, of course, detect and collate the similarities. So the computer will show the best matches. And then you are left to decide whether you want to further look for other ways to verify the results. So compared to a dichotomous key, polyclave keys are not only limited to a single or few characters. In fact, as long as the character states are known, all of them will be used to identify the plant. Now, this is very useful when you have, for example, a specimen that do not have flowers, and the dichotomous key available at the moment heavily relies on the reproductive parts of the plant. So it is more likely that your dichotomous key will be rendered useless in this situation. Now, a polyclave key do not only use floral characters, but also the roots, the stems, leaves. Okay, there are plenty of characters that is being used by this polyclave key. And it will also present a variety of potential alternatives in the matching. Okay, even if your specimen was not directly identified, you will have alternative suggestions. Okay, shall we say suggestions. Now, in a dichotomous key, once you reach the end of the couplets and you still haven't identified your sample, then there's nothing more to consider useful. And of course, your unknown remains an unknown. Of course, polyclave keys have at least, or shall we say, can present a few lesser matches for you to consider. And this minimizes the potential tedious process of starting back from scratch. For example, if your specimen happens to belong, for example, to another family, and you need to start again looking for a new key for that family. So there are already suggestions if you use the polyclave key. Okay, now let's look again at this online dichotomous key from the University of Michigan site. What are the things that we have discussed so far that you can notice here? And on the other hand, this is what I have come into online for a polyclave key. Okay, just check this out. Okay, it's kind of amazing how this subject made us discover a lot of plant systematics website. So again, we just check some characteristics here to limit or narrow the search until we get our desired species. Alright, now let's have written descriptions as another way to identify plants. And maybe everyone will find this boring or impractical, but if you're a plant enthusiast or scientist, you should always, or you should not ignore all possible sources of data for plant identification. Now, written descriptions can help narrow down the possibilities. Okay, you'll find plenty of this in various journal articles that tackle specific taxa of interest. Now, another method, of course, is to just compare your specimen with a known specimen. So whether it's a preserved one or a live one, as long as it's reliably and validly described. So it's easy. You just hold them side by side and compare them. But of course, what if they don't match? You probably will run out of collections with your unknown, still unidentifiable. And there is what they call here as synoptic collections, which house generally one specimen of each taxon for any given region or country. They are very useful if they happen to be present. But then, okay, you realize that you're here in the Philippines, so don't expect much. You are lucky if the plant that can be used to identify your plant is present in the given collection of the specimens. But still, you have to ensure that the reference plant was not erroneously identified, or else you'll end up identifying your unknown incorrectly. Okay, now you might ask, sir, why not just compare your specimen with pictures? There is a problem with that, with using pictures, because photographs and illustrations are usually available only for a small subset of your possible taxa. And in addition, it may be hard or complicated to locate the matching photograph or illustration. I think this makes sense because, of course, now you have an unknown plant. Well, what picture will you get to compare it? Where will you get them? Okay, so this is kind of ridiculous. But here, I came into this app, okay, called Picture This. Okay, just upload your pictures and then it will be identified. Okay, now technology overpowers all the difficulties stated in textbooks. Please check this out. So if it really works, then you can comment in the description below. And this video clip that I got from YouTube, credits to the owner. It showed here that it can indeed identify plants. And lastly, there are experts who can identify your specimens. However, take note that experts will still rely on the known protocols of plant identification. They can't do the identification instantly, okay, most of the time, especially for plants that you suspect to be a new species. And you also need to send them your specimen. So it takes time. You wait until they can allocate their precious time in helping you identify it. Normally, their expertise covers plants within a particular region or country. Some are experts only on some taxa, some specific taxa, maybe because they are doing studies on them, or they are the primary authorities in various botanical publications. But all in all, you should still be directing the identification of the plant that you need. So if you feel that you need an expert, then it's your call. So that's it again for plant identification, guys. You can choose to be practical in your methods or approach depending on the complexity at hand. Of course, you can try to use all of the methods that I have mentioned so far to triangulate things, because again, it's common that the identification is considered tentative unless verified by another method. Now, one interesting possibility is, of course, the idea that maybe your specimen is new, or it represents a new taxon. So, start thinking about Latinizing your name for this purpose of introducing a new plant in literature. So that will be our next topic for part 3 on plant nomenclature. And again, this is Professor Bryan Yves Araneta. Thank you very much for watching.